Hello, everyone, and welcome to today's AAPS Science of Compliance. Today's Science of Compliance is hosted by Somru BioScience. You may submit your questions at any time throughout the presentation by entering them into the Q&A box of the Zoom platform. All questions will be answered at the end of the program. It is my pleasure to introduce today's moderator, Jesse Ricks, Bioanalytical Principal Investigator for Somru. Jesse, the floor is yours.
Thank you, Stacy. And welcome to today's webinar on AI-driven compliance, harnessing machine learning from regulatory excellence. My name is Jesse Ricks, and I'm a bioanalytical principal investigator at Somru BioScience, where I've been for over 10 years. Throughout my career, I've been deeply involved in bioanalytical method development, validation, and regulatory compliance. And I've been excited to explore how AI and machine learning are transforming our quality management systems in our field. So before we jump right in, I want to thank you all for joining us. Today's session will focus on how AI-driven quality management systems can enhance regulatory compliance through real-time insights, predictive analysis, and automated decision-making. We'll also examine case studies, including ICH-M10 compliance, immunogenicity cut point analysis, and data integrity monitoring to understand the practical applications of AI in bioanalysis. Finally, we'll discuss actionable strategies for integrating AI into existing quality systems and optimize operations and streamline compliant processes. We'll be going over 3 case studies that showcase real-world applications of AI and regulatory compliance. After the presentations, we'll open the floor to Q&A, so feel free to drop your questions in the chat throughout the session. Now, I'm pleased to introduce our first speaker today. Mohammed Moin is the co-founder and CEO of Somru BioScience, driving the company's strategic growth and operations. An award-winning entrepreneur, Mohammed Moin has over 15 years of experience in healthcare and technology, with expertise in executive management, business development. Previously, he served as a solution architect at Acrion and solution architect at Maximus Canada, specializing in healthcare technology and drug information systems. Recognized for innovation, Mohammed Moin has received the Synapse Inspiring Young Innovator Awards and the E&Y Entrepreneur of the Year Award in Health Life Science. Now I'll hand it over to our first speaker. Please join me in welcoming Mohammed Moin. Moin, you're mute.
You're on mute. Oh, thank you.
There you go.
Thanks, Jesse. Welcome, everyone. A quick corporate overview about Somru BioScience. Somru was founded in 2012 in beautiful Prince Edward Island, Canada, a great place for your summer vacation. So we are a science-first, AI-driven digital CRO. We have been in the journey of digital CRO for the last 10 years. We built our own laboratory information system, LIMS, called AGRIS, and then for the last 2 years, we are developing the Gen AI technology on top of our AGRIS that we will speak about how we were utilizing it. We have about 30 plus molecule experience in the last 12 years, and we have been serving 150 plus biopharma companies worldwide. The next one. So what is Gen AI? The generative AI, the Gen AI is the buzzword going around, and every industry is trying to incorporate Gen AI. So generative AI is an artificial intelligence that creates new content, ideas, including conversation, text, image, or video from user input by learning patterns from the data and predicting what comes next. Gen AI is powered by large models that are pre-trained on vast amount of data, commonly referred as foundation model. The explosion of data in life science is driving the need for digital transformation, and I think we are at the life science industry is the cusp of utilizing Gen AI for their growth. So next slide. So what is the challenge? Every industry is utilizing Gen AI, but for the bioanalysis, we see there is a huge challenge in utilizing the Gen AI. Historically, all the data architecture, all the system that is used by CROs or biopharma clients, these are all closed systems. So all this, all the technologies, all the instruments in your lab that is capturing data, it is keeping this data in silos. There is no easily available interface to share data between multiple systems. The closed systems vendor, they don't share their data architecture, they don't share their insights. So there is a very lack of small amount of SME available, who can translate the data, understand the meaning of the data, the architecture behind it, and share the data between multiple systems. And also there is a challenge of regulatory demand on this. So because of these challenges, we see slow adoption of Gen AI in bioanalysis. Next slide. So how did Somru started building this Gen AI solution on top of Agris? So if I go from bottom to top, so this Agris cloud system can reside in any cloud. So it could be in Google Cloud, or it could be in your Amazon AWS solution, and it uses GPU and TPU. So we prefer, based on the client's preference on the cloud system, cloud infrastructure, we can go with any of the standard cloud infrastructure. On top of that, we built pre-trained model. So using Gemini, ChatGPT, and Claude, so these three foundational model, we have been tuning our own model and pre-training with the Somru generated data, publicly available data, and already curating all the data that is applicable for bioanalysis purpose. On top of this, we have called RAG, is a retrieval augmented generation. So this is where we plug in a client specific data. So this is your private information. This is, separately trains our model, but it is separate, specific to client A, B, or C. And how do we interface with these legacy systems, multiple systems you have, that we will take a fit for purpose approach, and we will look into our open interface data loader. So we have an open interface data loader that we have built that can pretty much gather data from any closed system in various different ways, and then it fits into our RAG system to train the model again for your own purpose. And on top of it, we provide the search and conversation API, the way you see in the chat GPT model, that it provides search and conversation capabilities. So our model has this search and conversation capabilities. On top of it, we have a multi-domain agent manager. So for various domain specific tasks, we have a domain specific agent that is being generated, and we wrap everything in on top of Agris system. So if we go to the next slide again. So Gen AI solution that we put on top of Agris LIM system, which we utilize as an expert system, it is as an LLM-based AI agent that will automate the manual, error-prone, time-sensitive data processing, analysis, and review. So it will use as an AI assistant for you on continuously giving you feedback on various domain-specific tasks. It is pre-trained with Somru generated data, curated data, and then it also gets regular feedback loop through your own data, that your own SOPs, your own guideline that your company follows, and then it keeps continuously training the model to give you better feedback. And it includes also various rule of thumb used by human expert, which is not possible to put into a rule-based engine. So this is where the differentiation from a rules-based engine to an LLM-based AI agent. It can also, based on all the feedback and stuff, it can also predict, the model can also predict and alert user before any potential issue can happen. So if we go to the next slide. So here is a high level, what are the type of agents that we use Agris to help you make better decisions, right? So the first one is the regulatory agent. So the regulatory agent utilizes all the approved documents, SOP, policy guidelines, your internal val plan, your SAP plan, report, training, all the information that you provide to us through our data loader to the reg systems. It can get trained on it and provide you feedback on regulatory. So my next speaker, Rafik, will present you a few cases that how we are doing regulatory agent and how the regulatory agent provides user the feedback. Other than regulatory agents, we have two other agents. We are actually hoping that by the end of this year we will have 10 different agents, but we have right now commercialized 3 different agents. So after regulatory agents, we develop a method development agent. As you all know, the pain of developing methods, you go through so many different iterations, and by 1 month or 30, 40, 50 days into your method development, you forget, okay, what was my essay number 5 or 10 or 8? Here, a lot of the people also in the past uses DOE-driven approach, but with AI agent, it accelerates your work and provides you a very optimized result. So your method development and your asset troubleshooting, So if all your asset related data is consolidated within Agris system, if you're using Agris electronic lab notebook systems, then Agris knows all the details. If it doesn't, then your ELN, we will capture the data through our OpenAI system overnight, and the next day when you try to plan for a experiment, the AI agent, method development agent, will be able to give you very informed decision that, okay, what needs to be tweaked in your next experiment. So that way you narrow down and troubleshoot your experiment and get to your final experiment very quickly, instead of trying hundreds of different times different parameters. It helps you decide what is the best option for your essay. And then we have a knowledge base agent that does continuous monitoring, trending, expiring, provides a predictive analytics for your task. Any of the tasks that you're doing, it will in the background, it will keep providing you real-time feedback as required. So we can go to the next slide. The Agris system can be used as a standalone system. So as a standalone system, when Agris is used as a validated cloud solution, so we pre-validate a lot of the things. IQ, OQ is pre-validated by Sombrew. PQ is done by a client specific to their need. It streamlines all your data analysis and report generation. So we have a separate module. So if you want to do just the immunogenicity work with Agris, you can do the immunogenicity work, immunogenicity calculation, method development, immunogenicity cut point calculation, things like this you can do within the immunogenicity module. Then we have a sample analysis module, biomarker, PK. So specific task you can do through a specific module. So you don't have to take license of entire software. You can pick and choose separate module. It's very intuitive user interface. It streamlines your data generation and everything. It has built-in sample and reagent lifecycle management. It is 21 CFR Part 11 compliant. And then on top of this product, for last 2 years, we are building the Gen AI solution on top of it. So this Gen AI solution becomes a AI assistant for your individual modules. So you could technically say that, okay, we want to take one module from Agris system and want to try it out. Or you can just say that, okay, I just want to utilize Gen AI features only, and I already have my own LIMS system and other system provide me Gen AI. So we can Agris as a Gen AI standalone solution can be also utilized by our client. The next slide. I think that's all I have for now. I will pass it back to Jesse for our introduction to next speaker.
Yes, thank you. That was great. So next, I'm pleased to announce Rafiq Islam, co-founder and president of Biopharma Development Services at Sommer BioScience. With over 25 years of industry experience, Rafiq has led scientific and regulatory initiatives at top CROs, including LabCorp, Celerion, Amador Bioscience, and Pharmaron. He is recognized expert in the bioanalytical innovation and regulatory strategy, known for developing cutting-edge solutions to complex challenges. As a reminder, please feel free to drop your questions in the chat. We'll have a Q&A at the end, and please join me in welcoming Rafiq.
Thanks, JC. I'll try to share my screen. So Let me see if I can. Makes it a little easier for me to present. Jesse, can you see my screen?
Yep, it's good. Thank you.
Thanks, Jesse, for the introduction. First of all, I apologize for my voice. Some of you probably know this is not my normal voice. I'm just recovering from a cold, and I'll do my best to power through this. I do have a pretty ambitious agenda here. Would like to cover three case studies next 20-25 minutes, and would love to leave at least 15 to 20 minutes for any questions. On the first case study, we had a sponsor as part of an asset acquisition. They had a PK method development, validation report, sample analysis report, and they wanted us to evaluate the quality of those results for both validation and also for sample analysis. They also have need, it's a small to mid-sized company with a lot of exciting asset, and they also had need for a solution where they can monitor multiple CROs performing those work. As you can imagine, in our line, you have, if you acquire some asset and they may have a method with lab A, and then another one has with lab B, it's not sometimes easy to transfer those methods. So when you find yourself as a small to mid-sized biotech company managing multiple CROs, multiple methods, they were looking for a solution where they can review those val plan report data more easily and be able to make a conclusion on the compliance risks. So in those cases, and Moeen covered on that architecture of our agri system, some of it is more rule-based, but what really gets me excited actually is that we actually passed that rule-based over the last 2 years and moved toward more in the deep learning areas where we can have the system provide some expert opinion and the expert recommendation and be able to identify gaps and the risk. At a basic level for validation, what it does is that it goes through 11 different attributes and checks again ICH M10, but also your SOP. I wanted to share a few screenshots. I know that these are not very readable, but I thought I'll just share with you just to show you in an architecture is how it looks. It's pretty much similar to Gen AI, but what differentiates is that we have a domain model that is built for specifically for PK, for immunogenicity. In this case, you have also option to upload your own SOPs if you already not using our full system and just using our only the AI module. You can upload those systems and be able to get a table clearly talks about where the gaps, if it is high, low, mid risk, recommendation for mitigating those risks, but also gives you specific document area where the section where you are deviating from or at risk of deviating maybe. Just wanted to share with you few of the finding. Obviously, for this specific case study, I had to remove some of the information just because it's confidentiality and when you are working as a CRO. But I pulled few different of those findings from different studies just to give you a flavor of the type of things that the system is verifying and looking at it, and some of the things that is providing recommendations on. So in this case, for example, blank metrics, it looks for all the description, lot number, receipt dates, all of those are there. Verifying calibration curve, not just for suitability of the curve, the different levels, number of points, but also the storage condition. And in this case, it did find a storage condition mismatch. And you can actually go into the link here and be able to find out where that mismatch is and be able to address those. In some cases, it could be just a documentation error you can support with your original raw data and be able to mitigate those risks. Sample tracking, for example, in this case, it found some of the duplicate subject. It looked at the total duration of sample collection to the last analysis, and there has been mismatch with the stability. If there is any deviation that is open, it can actually be able to identify that, the report is signed, you should not have any deviation open. Obviously, in this case, we will need access to your QMS system, assuming that the deviations are electronic. And a lot of cases, I think Moin touched on it a little bit, is that you can actually, a lot of the software vendor now allow for interfacing directly. In other cases, you can do an overnight export, and that's an automated setup. What really, I mean, truly, and I've been in the industry now 25 years, and I can tell you that spent out of those 25, 20 years in CRO industry, and a lot of my CRO colleagues here can attest to that, how complicated it becomes as you close a study and, tying up all the loose ends and, you find memo somewhere, some email, some audits, all of those needs to be closed. What really excites with AI is that, you can actually make it as big as you want. So in this case, I wanted to illustrate some examples where we gave our AI agent access to the email. We gave access to the QA audits. We gave access to a folder where we keep client memo. There are a few things that, it was able to identify. One of them is the dilutional linearity, where the report does not say, if you look at ICH M10, it says that you have to do 3 independent dilution series. And it did not find any evidence of the independence. Then the report did not clearly say that those series were performed independently. Selectivity, in this case, Valplan did say that selectivity will be done at low QC level, but M10 requires it should be done at LLOQ. It did find a memo that justifies that why being done at LQC level instead of LLOQ, but it's still identified as a risk and said in the report there should be some justification instead of a memo sitting in another silos. You want to include it in the report, the whole memo itself, or you want to put some justification in terms of why performing the selectivity for the hemolyze metrics at LQC level instead of LLOQ is still sufficient. Communication, some of the sponsor email communication, and again, the system can be developed to be as sensitive as you want. For example, if you have some email that's sponsor extended a reference standard expiry date through an email, that will be a critical email. With the model that we develop, it able to sift through the emails and be able to identify the critical emails that recommended for the report. Audit and inspection, same thing. The report is signed, but you want to make sure that all the audit were signed by the TSM. I believe in this case it was signed by QA or the BPI, but wasn't signed by TSM. At least one of the signature was missing, and it was able to identify that. In terms of some of the sample analysis, it does have, and again, I think some of the signs we try to make more, warning or green for is fine, or the red where you need a little more attention. On this one, it was mostly focused on some QC trend analysis. ICH-M10 encourage it. It's not required, but it was able to, flag it that, doing some trend analysis, also looking into the study concentration table and sample receipt table. There was some mismatch, and I believe in this case there was some aliquot number mismatch. There was some instrument ID typo that was able to capture through the system. Deviation from procedures, there was some open deviation. Again, it looks for all the signatures being done and it's being closed. It can go even as far as looking at the deviation and looking at the description of deviation. So most of us, we don't put everything that we put in the deviation in our report. You put an abbreviated comments or abbreviated explanation of the deviation and impact, and it's actually able to verify that what is written in the deviation itself and what is written in the report, they are consistent. Excuse me. When it comes to repeat analysis, it's identify multiple values. And again, in this case, most of the situation was that there was a repeat table, but it was not clearly identified, or the report discussion did not really clearly. So it was not truly multiple acceptable values, but there was multiple values that were in a table, and it was able to flag it. For ISR, for these specific scenarios, ISR did pass. However, the SOP required that any ISR sample fail greater than 50% needs some resolution. And again, this is this is not something practice we follow, but in some cases, if you have a company practice in your SOP, you mentioned that our system is able to capture that and be able to suggest to the PI to be able to follow those SOPs. And this one really we did the last one on the inadequate calibration and QC range. We have one of our assays, free IgE assay, and we actually wanted to see if the system able to capture that when you're measuring free IgE, the level is so low, our QCs were on the top, and we did change the QCs, but we wanted to see that if AI system is able to capture and make that recommendation. And when we put in the data that were initial data that was run, where you have a pretty wide calibration curve, but the samples were falling right on the below, and you had only one QC that covers that range, and it is a requirement in the ICH M10, the AI system able to capture that and be able to recommend that you need some scientific justification and discuss the impact on the report. Excuse me. So in summary, I think, it's a couple of points. I don't know, if you are looking for more retrospective type of analysis where you have the plan report, more like, as a CRO, but also as a biotech company where you're being asked to review a plan report, val plan or validation report or sample analysis report. This is something pretty quickly you can review. In some cases, we have seen a significant decrease, up to 50% decrease in the time required. Can reduce a lot of reduction in rework in case of your QCs falling, not falling within the sample range. It helps you identify it early on, makes the correction so you can actually update it. And definitely overall, it enhances your regulatory compliance. I'm just going to switch gear a little bit here on the immunogenicity. In this case, we have a biotech company in the area of gene therapy doing some immunogenicity assay where there is a pre-existing antibodies are there, and what we were trying to solve in terms of the validation and mostly focus on the really getting the right cut point here. And then, one of the things we're working with them also, the developing a model for integrated summary of immunogenicity. Pretty much anybody who have done immunogenicity is familiar with this chart from Dev Narayan paper. It's pretty well utilized. I wouldn't go into details of it, but in a flow chart, on its face value, it's pretty routine, really. I mean, you do some normality test, and it's parametric or non-parametric, and then you follow parametric approach or non-parametric approach for doing cut point. But then if you are like me in the industry for almost 20, 25 years, once in a while, 1 in a 10 cut point, you get into the situation where you have quite a few of complex situation going on where you do the cut point one way is too high or too low. Frankly, like my buddy on this picture, feel like you're going to pull your hair out. So in this case, we had a data that is not normal even after the log transformation. We have some high CV, and some of it was really more pre-existing antibody. But then by doing some confirmatory, we were able to remove that pre-existing antibodies from the calculation. But you always get that low level of pre-existing antibody that does not meet the confirmatory threshold, and you don't have any justification to remove them. And if you do non-parametric, the cut point is too high. You can see that you're losing your sensitivity at 100 nanogram. You're getting a lot more false negative. If you do parametric, sometimes it's too low. And I think in this case, we tried different ways of doing parametric with removing high CV samples. And you end up with too high or too low cut point. And what we did with our immunogenicity here is that, we looked at, went through pretty step-by-step process, that is being described pretty well in the industry. And we did all the tests you can think of, all the statistical tests in terms of normality test, Levine test, ANOVA. And it did identify that you want to have a floating cut point. We have some concern about some of the samples was a little high CV, but if you look at some of the low response, you can really apply CV to remove those low response, then you're getting your cut point a little too high. In this case, and again, I know you can't read that, so I'm going to go to the next one. But what I wanted to show in terms of that, in addition to doing the statistical analysis, you have that expert assistant. You can ask the question, and in this case, it made a recommendation on how to proceed with this cut point. And the recommendation was that drop one of the assay because of the high variability. The variances are not, once we remove that one assay out of 6, the variances were no longer significantly different. We use so even though the normality test did not pass, the skewness and the kurtosis values were within range that we could use a parametric approach, but more robust parametric approach instead of mean-based approach to median and MAD. And then perform, also it recommended that, to perform in-study cut point as soon as the in-study samples are available. And, a lot of these things that we could come to the same conclusion. If I have an expert statistician, an expert immunogenicity scientist together, we can come to that conclusion, but it will probably take a few weeks to get to that conclusion, putting all the data together and going through this process. But in this case, we were able to get to that conclusion in a matter of hours, really. On this one, just wanted to thought I would share with you some one of the things that we have under development, and on the right-hand side, that knowledge base, that's really more Moin's area. On the left-hand side is my area. What we're looking at is that creating 4 different domain knowledge for supporting integrated summary of immunogenicity. Anybody who worked on it, on my last one, we worked on it. We have like 3 people, maybe, including our data management team. You have close to 8 to 10 people working 3 to 6 months to come up with the integrated summary of immunogenicity for an NDA submission. And a lot of those information are already in the different clinical report, protocol, CSRs, and assay validation, sample analysis report. Those CMC documentation, they exist. And for us is that how we can develop an AI model to make this thing easy for our scientists. So you are not going through those thousands of documents to put together your ISI. And we have shown some significant promise on that, and we expect to release that by end of Q2. So in summary, when we did the immunogenicity using that AI-driven cut point approach, in addition to those statistical process, we were able to get to the conclusion pretty quickly. And it's not only that you get to the conclusion, but it's not, even though AI itself, it's somewhat black box, But the answer is not black box. It provides you the references, scientific justification. There's something you can put in the report to justify the approach you took for your cut point. So it definitely enhanced compliance and definitely also improve the sponsor confidence. The last but not least, where ultimate goal is that where we want to go. I'll skip the other slide. So our ultimate goal is that we want to get into real-time compliance monitoring. The two example I showed you is almost like, it's retrospective. You let people make error and, a lot of different, generate more challenges for yourself, and then you go and solve it. So how can we really prevent, those issues from arising in first place? And the way to do it is that having an AI model, AI agent that can do pretty much real-time monitoring of the data integrity. It can, prevent error before it can occur, before it can happen, reduce bottleneck, and act as an trusted expert SME, almost like having an expert friend next to you who can help you in terms of guide you through the decision making and compliance. So in this case, some of the things we're looking into is that automated error prevention, real-time monitoring, continuously checking for deviation in critical parameters, doing some predictive modeling in terms of trending, and standardizing QC workflows. And I'll share with you some, couple of small examples just in the interest of time. One good example is that, in this case, for this study, we have and I'm sorry, you cannot read it. It's just a screenshot from our software. In this case, we had a dilutional linearity that is established up to 50-fold. The analyst here entered 60-fold, and right away it gave a warning that you can only, it is outside the limit. You can only dilute your samples up to 50-fold, but also gave you a recommendation. So as you are analyzing samples, in most cases, if you have a linear dose response, and even if it is not linear, this is what deep learning is good at, identifying patterns. So if you have already analyzed 20 subjects of the 600 subject clinical trial, it can start predict what your dilution should be for each time point. So, I have my BPI's days where I'm pulling my hair and I feel like I, diluted too much or too little. It's nice to have a tool that is using some of this predictive modeling to suggest what should be the right dilution. So it's not waiting for you to make mistake and then telling you. In another case, what we did here, you see in an assay, we have couple of QCs. I think in this case, one QC is outside the range. And we set up an alert limit if it is greater than 3 sigma. And in this case, it was able to identify and say, okay, here is an alert for you. And it also looks into SHAP values. And, SHAP values is basically showing that which are the factors that are more significant in this case. So in this case, coding protein, I can't even read this so small, but I think coding protein substrate, and I think the kit lot was some of the significant. Some of them has the positive. I think the significant relationship was coding protein on the positive side of 0.7. And that allow you to go into more of a more detailed investigation. Now, if it is a one-off, it happens only one time out of 20 times. You don't need an investigation, but it still is good reminder to keep an eye on. But if it happens, multiple times, definitely this you can initiate an investigation, and the AI tool able to support you through the trending and providing the data for making those decisions in terms of the impact on the existing assay or how to prevent it from seeing repetitive or systemic error that might fail your assay in near future. So some of the few of those predictive modeling that can help more real-time monitoring of compliance is the monitoring ISR performance, real-time carryover check. We have actually a domain knowledge that continuously checking for your plate data to be able to alert you if there is any carryover suspected. suspected. Adaptive rerouting of samples due to dilution or anomalous value. If you have some SOPs, and again, keep in mind that AI does not decide it. So a lot of it is driven by your SOP, how you prioritize your processes and the documents within the system. It can, if you do PK repeats for the anomalous value, you can recommend those ones. If you are off because of the dilution, it can automatically, or with user input, can reroute those sample in an adaptive way. Also, adaptive QC trend analysis to detect any systemic error, any sort of escalation process for decision making that also can be automated using this real-time monitoring system. So I wanted to close with just some thoughts in terms of what is the best way in a bioanalytical lab to integrate that AI ML. One thing we learn is that when we're working with some of our clients, the first thing is that the AI readiness. A lot of times the SOPs and all the information data is not in a digitized form, but if it is in digitized form, they are in silos. Some of excuse me, some of the old school software does not allow you to really integrate easily. In those cases, we develop tools where you can export all your data overnight, and then the AI agent can sort through all this data and be able to help with the decision making process. Other things that moving toward a standardized technology, terminologies, I should say, that is consistent with global regulatory guidelines. I mean, if you call matrix effect versus selectivity, it's good to have some sort of nomenclature terminologies that are consistent with the global regulatory guidelines. For some of the methods where there is not a well-accepted guidelines or the industry standard, we recommend our client, you can write a white paper for your company. You can write SOPs. SOP is not probably the best way to add more SOP, but having some sort of guideline white paper where you can pull from between different industry white paper maybe, or if it's possible that WRIB has a lot of great white paper, you just follow them and we can use those. They are already actually part of our SOMRU model, and you can utilize those as a standard for verifying your assay performance. The investing in AI training, upskilling for bioanalytical scientists is, I know my early days when I'm, not really very comfortable in statistics, being a biological scientist, but AI is here to stay, and I think it's important for investing for the companies in training and upskilling. And last but not least, I mean, really needs partnership, and I think what we do well here is that I am more of an user of the tool, but we have Moin and a whole team here who actually collaborate together to get to this finish line. I mean, pretty much everybody in the company is really one way or another involved with this. I do wanted to mention a few names, Joey, who is the product manager, Al-Walid, one of the lead application developer, Jesse, Will, our PPIs, Clarinda, who manages as a CEO of the company. And we also I want to recognize the University of Prince Edward Island, our collaboration with them for years and years. We've been working together here to develop some of these novel tools. So thank you very much for your attention, and I'll pass it back to you, Jesse.
Thank you. So thank you to both of our speakers. As we've seen today, AI is increasingly relevant in regulatory compliance, particularly in bioanalytical studies and software applications. I can say, as a principal investigator, I do see firsthand how AI-driven tools like Agris make a real difference. They boost data integrity, simplify compliance, and help us make better decisions. And in the end, they actually cut down on the paperwork and allow us scientists to deal with more science. So for those interested, we invite you to pre-schedule a demo at the WRIB and the AAPS conferences that are upcoming. And now I think we can get to some questions. If you haven't dropped one in the chat already, you can continue. And if we don't get to your question during our Q&A, we will have a list of the questions and the emails, and we will respond to you via email in the coming weeks. So getting to our chat here. Sorry, just going to move. The first question that I have is, do we need to validate the AI agents? I think this goes to Mohammed Moin.
Thank you for the questions. Yeah, so AI agent validation, it depends on the context of your use. Agris as a solution is being validated for various modules. So if you're using immunogenicity module, PK modules, or our sample analysis module, the systems are validated, and then client does their fit for purpose validation. But AI system, the AI assistance, which is part of immunogenicity or PK or other modules, you do not need to validate it because it's not making a decision for you. It's helping you to make a decision. So ultimately, the bioanalytical principal investigator is making the decision. So it depends on the bioanalytical principal investigator to whether to accept the suggestion or not accept it, and then provide the feedback back to the AI agent. So right now it is a research use, so we are not validating it, but we are looking into regulation. For example, FDA has come up with the guideline and draft guideline, and is receiving feedback from industry when, in what cases that AI model needs to be validated, and there is a greater discussions going on. But ultimately, it depends on the context of the use.
Yeah, I can add maybe simplified way of saying I have 3 case studies. If you're doing case study 1, just more of an information, you don't need to do any validation at all. Case study 2, you do need to do a little bit of qualification, PQ, and case study 3, where you are really making decision. In some cases, you are letting AI make the decision for you. If that is the case, absolutely, you'll need to fully validate in those cases. So there's 3 different scenarios, and it's really dependent on how you're using the tool to make any regulatory decision.
All right, thank you. Great question. So the next question I have, I think, goes to Rafiq, as we're talking about case study 2. So in case study 2, how does the AI agent determine the optimal cut point method for immunogenicity assays? And can it adapt in real time based on new data?
Yeah, so one thing, and again, maybe I can just quick share my screen again. One thing we could not, didn't want to bombard you guys with a lot of screenshots. So basically what it does is that, it calculates cut point multiple different ways. So what you can see on this one, I think, behind this screen, you have like 1, 2, 3, 4, 5, 6, 7, 8. So it calculated 8 different cut points. So what it's looking for really, what your SOP, what your val plan, what is the industry white papers are out there. And again, AI in some way is a black box, and this is why I think regulators are also trying to figure it out. What is the best way to validate those tools, really? And we can do we are doing a white paper on it. We can do a whole another presentation on those. But the idea is that it is taking the cue from your SOPs, like, SOP and the industry white paper will say you look into the variability between the 6 data set that you generate. You can remove one if you can see that your overall variability decreases. I think you can remove at maximum 2 out of that 6 data set. But if your SOP says you cannot remove the data set, you wouldn't do it really. So it will go through all the acceptable path for cut point based on your plan, based on what the publication out there, and then identify the cut point that is truly statistically. Our goal here is that generates only 5% false positive. So in some way, it's doing what human does, but if I were to do 8 different ways, the cut point, and then evaluate, and then make a decision, it will take me if weeks, if not month, maybe, and will require multiple domain knowledge expertise in those conversation. And in this case of AI, it's really sifting through all these different case studies even out there in the literature, and based on your SOP, and be able to identify a cut point factor that will give you that closer to that 5% false positive rate you're looking for.
Okay,
I hope
great.
this
Thank
answered the
you.
question. Yeah.
I think so. So moving on, next question I have here. How does the Agris AI-driven compliance system compare to traditional LIMS solutions? Can it function as a standalone compliance tool, or does it require integration with existing lab infrastructure?
I can take it, Jesse.
Oh.
So it can be used as a standalone tool. So if a CRO, a lab already has their LIMS system, and already has all other lab informatics solution in place, and they want to use it as a standalone regulatory agent, it can be used like that. In that case, there is an analysis of their systems we have to do, and then we utilize our open data framework that connects with the various systems. Not all systems allows us to connect directly. So instead of active integration, we do a passive integration, overnight data loads and dump into our system, so that the system gets trained on their specific data on top of our pre-trained model. And after that, they can use like a conversational style.
Okay, that makes sense. Thank you. So then let me see here in the chat. If we implement as standalone agent, what does the implementation look like, and how long would that take?
So it depends on, we will do an exploratory meeting with the client to figure out what are the list of systems they are currently utilizing to capture data. So depending on their system list of electronic system they have, and list of interfaces we need to build using our open interface, it depends on that. But I can say that usually it could take from 3 to 6 months for an implementation.
Okay.
Yeah, I mean, unless you're using, I guess, or off-the-shelf model for more exploratory purpose, that can be done as little as 2 to 3 weeks, really. So it's really dependent on how you utilize it. If it is more of a exploratory thing as opposed to a decision-making tool.
That makes sense. Okay, thank you. So then the next question that I have here, the regulatory guidances for technologies such as flow and qPCR are not as standardized. How would AI be able to help?
No, so this is a good question. I touched on it on my presentation a little bit. It's something we are trying right now. And one approach we took is that we started writing some white papers. It's a little more than your SOP. See, SOP is a little more rigid. In a white paper, you can actually describe different ways. You can make references to different articles. And again, going to this the deep learning, that what really more exciting is that it's able to sift through different publication, but your white paper gets a higher weight on the decision making. So some of the initial model that we did, we do see a lot of promise, but this still needs to remain to be seen, that what level of details we can go, and what will be the level of utility we can get out of those model that focus more on those validation sample analysis for flow or qPCR type of methods.
Right, that makes sense. Okay, and then I have one other question here. What do you see as the biggest challenges in implementation of AI in bioanalysis?
When you want to take that, or you want me to?
I was passing to you, but yeah, we both can. You can take a step, and then
Yeah. I mean, I think there is maybe two. One is that, from a bioanalytical user perspective, um, is, you know, getting educated on it. I spend a lot of time with Moin and also our team here trying to understand, um, how it is, one of the questions I always ask that, how did it reach to that conclusion? And they will sometimes correct me. More important is that is the conclusion is correct. And, how it will require some continuous updating of the model to make it effective, because we work in a very dynamic environment where the regulation change. So how do you manage that? But even before having all the data, having a consistent data structure, again, good thing with AI is that you don't need to the level of like having a standardized database of data, because it can understand human language well. But at the same time, you cannot have conflicting type of publication and stuff in the model that can minimize the regulatory rigor that is required. So it's the data structure, is the some training, and how we keep those updated. And I see as actually very relevant question on this is that, do we need to revalidate the AI model as it continues to learn and adapt? The short answer is yes. Some sort of revalidation doesn't mean that you have to spend weeks and months revalidation. No, but if you are making a regulatory decision, and I was actually just read up the other day the AI guidance that came up from FDA for medical device, and there was another one came up for what was that? I think clinical pharmacology modeling. Um, and it goes to like a lot of us in bioanalytical community familiar with the biomarker validation and the context of use. So I'm afraid I have to bring that context of use again to this discussion, and it's going to depend on the context of use, but it will require some continuous monitoring, that's for sure.
So I can add my 2 cents here, Rafiq. So what, from the computer science point of view, what we don't like in the bioanalysis is so many maybe, so many rule of thumb the scientists used over the years. So the model takes continuous feedback, and that's why we have done a lot of tuning of the model, and that's why we have, started with multi-model design, right? Multi-domain specific models. So we use ChatGPT, Gemini, Claude, 3 different models where we utilizing, and we trained it, and we found that, okay, sometimes we do cross-model checks, right? So when you are trying to say that, okay, how often do we need to validate? So to reduce the burden of validation multiple times, what we are doing, we have some built-in checks. AI is known for hallucination, right? So how do we prevent in a regulated environment when you are using it? How do you prevent it from hallucination? So we have built-in checks, right? The answer gets checked multiple times through different models to say that, okay, AI is not hallucination. So it prevents it from giving it a false answer with very confidence, right? So if the answer is not 100% sure, the system knows that, okay, my reasoning process that I went through, and I have seen multiple contradictory information and stuff, and I'm not 100% sure, it will present you as an answer that, okay, you can tell that, okay, it's saying that, okay, these are the rationale I applied, but there is also conflicting solution information as out there to make a decision, allow him, the PI to make the decision instead of the system giving you a false answer. So to prevent the hallucination, we have built-in system in place.
Great, thank you. Those are some great answers. I think that actually ends us on a perfect timing. I think that is it for us. I see a couple more questions that we may be able to get to via email back to you. Anything else, Rafiq or Moin, you would like to add?
No, just
No, I
want to
think
thank you everyone for joining, and you have our contact information. I think there was a question about the recording that will be available on AAPS, and you have our contact, and feel free to reach out to us if you have any questions.
yeah, and feel free to schedule demo. The recording, the slide decks will be available. If you are not at the AAPS or WRIB, you can still request. Please follow our LinkedIn page. The links and everything will be available through our LinkedIn page from Somru Bioscience, and you can schedule a meeting with our team for a demo. Thank you very much for your time.
Thank you, everyone. Thanks, Jesse. Thank you.
Thank you.